This is Dr. Andalip Zehra. I'm an MBBS doctor, but I'm in teaching profession for last 23 years. I think teaching is my passion. I really enjoy teaching young students. In A level, I teach biology, but in O levels, I teach biology and chemistry both. Because I am teaching biology to both O levels as well as A levels, so I have opted to discuss syllabus requirements of A level as well as a topic which is covered in biology of both O level as well as A level. So the syllabus number in A level is 9700. And what CIE expect from the students, they check for their knowledge and understanding. And they have only one way to check both knowledge and understanding, and that is their papers. So the main focus comes on handling of information. Though sometimes students claim that they have the knowledge, they understand the things, but they don't know how to write down answers. Here comes the handling of information. So obviously, when we are doing our paper practice, we focus on the handling of information, means how to write down answers properly, because I feel appearing in an exam is an art. You have knowledge, but if you can't convey it to your examiner, means you won't be able to get marks. Then similarly, evaluating information, because in biology, this sometimes give you a detailed passage or a long data. You need to focus on that data, you need to analyze it, and then you need to write down the answers. This is usually, or it's better to say, it is more commonly seen in paper 5. Now, as I'm discussing papers, because this is the only assessment criteria of CAIE, they assess you in papers. So let's discuss these papers very, very briefly. Now, P1 is a multiple choice paper. It is for 1 hour and it has 40 MCQs. Then P2 is a structured question paper and it has 1 hour and 15 minutes. Paper 3 is for practical papers. It is for 2 hours and it usually have 2 questions here which you need to do. Then similarly, paper 4 is again for 2 hours and it is uh, again a question paper of structured questions. And P5 is for 1 hour and 15 minutes. It is for planning, analysis, and evaluation. A very, very important paper, because this is the paper in which they give you data you need to analyze and then answer the questions which they are asking for. Similarly, in other question, again, they give you a detailed information or data, and then they ask you to design an experiment how to reach a certain conclusion. Now, for today, I have selected the topic of photosynthesis because most of you are familiar with the process. You have done it in O level and you go further in the details of this topic of A level. Now, what is photosynthesis? By definition, it is a process in which green plants make their own food. How do they make their own food? They use sunlight, carbon dioxide, water, and other minerals to make glucose. And they convert light energy into chemical energy. Now, here in this paragraph, my main focus is on light energy, because you cannot say plants use solar energy. So term solar energy is considered as wrong, because solar energy has heat energy as well as light energy. So our focus must be on light energy. Now, equation for photosynthesis. There are 6 molecules of carbon dioxide. There are 6 molecules of carbon dioxide. Please follow my cursor. They react with 6 molecules of water in presence of sunlight, right? And they form one molecule of glucose, which is C6H12O6, and 6 molecules of oxygen. Now the important thing is that when you write down chemical equation, it must be balanced, because we cannot write down an unbalanced chemical equation. Let's say if you can't balance, then write down word equation instead of chemical equation. Then similarly at higher level, we may annotate the diagram a little bit. We may write down over here below arrow that a green pigment called chlorophyll is also needed. So in this way, you can write down complete equation by adding chlorophyll to it as well. Now sites where photosynthesis can take place. To all my O level as well as A level students, they all know that photosynthesis takes place in palisade mesophyll cells. It takes place, there are two different type of palisade mesophyll cells. There are spongy mesophyll cell, then there are column-like mesophyll cells, and at the same time photosynthesis also takes place in guard cells. If we simply focus on this diagram, we can see that these are palisade mesophyll cells, which are column-like. These are spongy mesophyll cells, which are irregular in shape. And if you follow my cursor here, you would be able to see, let me mark it very clearly. You can see these are guard cells where photosynthesis can take place. So now in this diagram, we are focusing on spongy mesophyll cell. So you can see that this cell is taken here and it is magnified here. Now, if we look in these cells, we would be able to see this structure. This one, this is a chloroplast. And you all know this, I have colored in orange. This is a chloroplast. If we further focus on the chloroplast, we would be able to see it is a sac-like structure. Till here you all have informations, and till here this is our O-level syllabus. But detailed structure of chloroplast is in A-level. So let's see what is the detailed structure of chloroplast. So the main organelle which is responsible for photosynthesis is chloroplast. Now please look here. It is surrounded by two layers of membranes. Membranes are exactly like cell surface membrane. Chemically, these all membranes are same, means the membranes which are forming cell surface as well as membranes which are forming outer boundary wall of chloroplast, chemically they are same. Now if we'll focus over here, this is the outermost membrane. So let me write down, this is outermost membrane, let me label is it as 1. Then you can see another labeling over here, this shows inner membrane. It means that it is made up of two membranes, outer membrane as well as inner membrane. Now there is a little space present between two membranes. What do we call it? We call it intermembranal space. So I have numbered outer membrane as number 1, inner membrane as number 2, and intermembranal space as number 3. Now inner membrane, you can see, please follow my pen. This inner membrane, you can see that it is thrown into folds. These are folds. These are basically fluid filled sacs present over here. And what do we call these stacks? These stacks are called thylakoid. Okay, you can see here they have labeled them as thylakoid. So these folded surfaces are called thylakoid. Now these are fluid filled spaces. I want to show you that how thylakoid, let's say this is the inner membrane. This paper in my hand represents the inner membrane. Now how thylakoid will be formed? If you look at it, you would be able to see this is how the membranes are present. This is, just a minute please. This is one folded membrane, you can see clearly. Then you can see another folded membrane. Then you can see another folded membrane. So this is how. Now I need you to understand this paper appears to be so small from every side you can see, but if I unfold all these layers, it will become so large. It means that inner membrane presents a very large surface area where photosynthesis can take place. And now each membrane which I folded and I showed you, this is number one, this is number two, this is number three, these are thylakoids. But these thylakoids, you can see here, even in this diagram, you can see very clearly that they are piled on the top of each other like stack of coins. Now this structure which form the stack of coin, this is known as granum. I will number it as number 5. This is granum. What is granum? You can see the labeling here. It is stack of thylakoids. Granum is singular. You can see how many such stacks present over here. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So plural of granum is grana. I will say there are 10 grana present in this chloroplast, and each and every granum is made up of several thylakoids. Now, what is this which is represented by yellowish color over here? This one, right? I am further highlighting it with yellow color. What is this? This is a jelly-like material which is found inside the chloroplast, and we call it stroma. It is a jelly-like material. I hope structure of chloroplast is clear to everyone. So now let's go to the structure related to function of chloroplast. Structure related to function of chloroplast means the structures which we have seen here, how they help to carry out in their functions. So on one side, I have named the structures and their details, and right in front of them, there are the functions of all these structures. Now the outer membrane, you just saw outer membrane. It is very, very smooth. So, and I said it is just like cell surface membrane. So what is its function? Its function is to control entry and exit of different chemicals. Similarly, coming to inner membranes. Inner membranes are thrown into fluid-filled space called thylakoid, which pile on each other. And each fold is called granum. Now, what is the function? You can see I have numbered 3 functions over here. They, the first function is that they provide attachment to photosynthetic pigments, means those pigments which help in photosynthesis, and you know one such pigment is called chlorophyll. If I will continue with this lecture, I will give you further detail of different varieties of photosynthetic pigment. Then similarly, these inner membranes, they provide attachment to stock particles. Now the question is that, again, this term is new for you. What is the meaning of stock particle? These are thumb pin shaped particles. We call them, I will draw one more. It will give you an idea. These are thumbprint shaped particles, and these particles are very, very important because they help in the formation of ATP molecule. I hope all of you are familiar with ATP molecules. ATP stands for adenosine triphosphate. This is an energy currency or energy molecule. So, second structure, yeah, second function of inner membrane. First function, they give attachment to photosynthetic pigment. Second function, let me write down, this is the first function of inner membrane. This is the second function. They provide attachment to stock particle which help in synthesis of ATP. And what is their function? They help in ATP formation. Now coming to third function, they, because I said these are folded membranes, when I unfolded my paper, I showed you how large surface area it is covering. So it provides a large surface area for photosynthesis. Now as a summary, I can write down that it is a site for light dependent stage of photosynthesis, because photosynthetic pigments, they trap light and they help in the synthesis. Partly they help in synthesis of glucose. So I can say only the light dependent stage takes place over here. Now I have discussed outer membrane, inner membrane, then coming to intermembranous space, which I told you over there. Intermembranous space act as a reservoir for hydrogen ions. It let hydrogen ions to be collected over there. Details and importance of hydrogen ion will be discussed in any upcoming lecture, but at the moment let me summarize it. Hydrogen ions also play a very, very important role in formation of ATP, which is needed in dark stage of photosynthesis. Even to my O-level students, you all know, this is your basic knowledge, that photosynthesis takes place in two stages: light stage and dark stage. So during light stage, ATP is formed which help in dark stage of photosynthesis. Now the last part of chloroplast which I will discuss is stroma. Stroma is a jelly-like material. Now if it is a jelly-like material, what does it contain? It contains, no doubt, what it contains. It contains, no doubt, water. It contains minerals without which glucose cannot be formed. It contains some very important organelles like ribosomes. Why? Because ribosomes help in enzyme formation which are needed for photosynthesis. So, yes, it contains one more important structure which I need to discuss over here. This is circular DNA. Now, why circular DNA is there? Because circular DNA give instructions to ribosomes to make enzymes. So it means that we can, with full confidence, we can say that stroma is a main site where important chemicals are made, and chloroplast is an independent organelle which can carry out all of its functions. So this is the list of the chemicals which I have mentioned over here, and it is a site for light independent stage of photosynthesis. Means now here ATP which is formed in light dependent stage is used to make glucose in presence of different enzymes. I hope it is clear to everyone. I want to keep this demo a bit short and interesting for all of you. So let's see. Because this is generally how I teach in class. Sometimes I find good video clippings to show my students that how. So here they are using an apparatus called photosynthesis, which will help you to find out that how photosynthesis is taking place and how what is the rate of photosynthesis. This video is without sound. That's why I am explaining along with the video. Now this is an aquatic plant. You must know the names of aquatic plants. It may be hydrilla. It is very important to maintain temperature, because if temperature will be high, as I have already discussed, this is an enzyme dependent reaction, so enzymes may get denatured. This is how they have attached a syringe to the capillary tube. In presence of hydrogen carbonate, it means it has plenty of carbon dioxide, so photosynthesis will take place. Here they are placing the end of water plant under funnel. You would be able to see this bubble coming out of the cut stem. And these bubbles will obviously accumulate in the glass syringe. So over a certain period of time, we would be able to collect certain volume of gas in syringe. You can see a bubble moving in it. So this is how the experiment is carried out, and I usually keep showing you different videos related to experimental activities or different animated video needed for it. So that's all about my today's lecture. Uh, Hope to see you in my class, and I think you will be in quite expert hands where you will learn a lot. Thank you very much.